medication for 1,700 days.
Disclaimer. The content on this channel is meant only to provide helpful insights, tips, and information on certain subject or topic. While we strive to provide accurate and up-to-date knowledge, this is not a substitute for professional advice. Always consult a qualified expert or professional for your specific needs. We encourage you to use this content as a guide for learning and additional information. If you think this does not fit what you want, please skip the video.
CRISPR is being explored as a powerful tool to treat cancer by making precise edits to the DNA of cells, especially immune cells, so they can better fight cancer. Cancer occurs when cells grow uncontrollably, and CRISPR allows scientists to reprogram how these cells behave or enhance the immune system's ability to target cancer cells. Here's how CRISPR is being used to treat cancer, explained in a detailed but simple way. Boosting the immune system with CAR-T therapy. One of the main ways CRISPR is being used in cancer treatment is through something called CAR-T cell therapy. This therapy involves modifying a person's own immune cells to make them better at recognizing and killing cancer cells. How it works? T cells. Our body's immune system has special cells called T cells that help fight infections and diseases, including cancer. But sometimes cancer cells manage to hide from these T cells, making it hard for the immune system to detect and destroy the cancer. Modifying T cells with CRISPR, scientists take T cells from a cancer patient's blood and use CRISPR to edit the DNA of these cells. CRISPR gives the T cells new instructions, essentially reprogramming them so that they can better recognize and attack cancer cells. Specifically, CRISPR is used to insert a chimeric antigen receptor, CAR, into the T cells. This CAR acts like a special GPS system that helps the T cells locate cancer cells. Reinfusing modified T cells. Once the T cells have been modified with CRISPR, they are grown in the lab to increase their numbers. These enhanced T cells are then infused back into the patient's body, where they seek out and kill the cancer cells. This approach has shown promising results in some cancers, like certain types of blood cancers, such as leukemia and lymphoma. The advantage of using CRISPR in this therapy is that it makes the T cells even more precise and effective at targeting the cancer cells. Disabling genes that help cancer grow. Cancer cells often thrive because of specific genes that promote their growth. CRISPR can be used to target and disable these cancer-causing genes. How it works. Finding cancer-causing genes. Every cell in our body contains genes that control how it grows, divides, and dies. In cancer cells, certain genes known as oncogenes go out of control and tell the cell to keep growing even when it shouldn't. CRISPR can be used to find and switch off these faulty genes. Cutting out the problematic gene. CRISPR is programmed to target a specific gene in the cancer cell's DNA. Once CRISPR locates this gene, it uses its molecular scissors, the Cas9 enzyme, to cut the gene which disrupts the faulty signals that make the cancer cell grow uncontrollably. By disabling or knocking out these oncogenes, CRISPR can slow down or stop the growth of cancer. This method is particularly exciting because it allows scientists to tackle cancer at its root by directly fixing the faulty genetic instructions that cause it. Editing the tumor's surrounding environment, also known as the tumor microenvironment. Cancer cells don't exist in isolation. They're surrounded by other cells and structures that can help them grow. This surrounding area is known as the tumor microenvironment. CRISPR can be used to edit the microenvironment to make it less friendly for cancer cells. How it works, removing protective cells. Some cells in the tumor microenvironment, like T regulatory cells, also called TREGs, actually protect the tumor from the immune system. CRISPR can be used to edit or remove these protective cells so the tumor becomes more vulnerable to attack by the immune system. Enhancing immune response. By using CRISPR to modify immune cells and the microenvironment, researchers can make the area around the tumor more hostile to cancer. This might involve boosting immune signals that attract more immune cells to attack the tumor, or cutting off signals that allow the tumor to grow. One of the biggest challenges in cancer treatment is that cancer cells often become resistant to traditional therapies like chemotherapy and radiation. CRISPR is being used to make cancer cells more sensitive to these treatments. Cancer cells sometimes develop resistance by mutating certain genes. CRISPR can be used to edit out these resistance genes, making the cancer cells more vulnerable to existing treatments. By disabling resistance mechanisms in the cancer cells, CRISPR makes it easier for these treatments to work more effectively. Personalized cancer therapy. Since each person's cancer can be genetically different, CRISPR offers the potential for personalized cancer treatments. With CRISPR, scientists can tailor treatments based on the specific genetic mutations present in a person's cancer. How it works. Analyzing tumor DNA. By sequencing the DNA of a patient's tumor, doctors can find the exact mutations driving the cancer. Customizing CRISPR edits. Once the mutations are identified, CRISPR can be customized to target those specific mutations. This makes treatment more precise and reduces the likelihood of harming healthy cells. This personalized approach has the potential to make cancer treatments much more effective and reduce side effects, since treatments would be designed specifically for each patient's cancer profile. Real-world examples of CRISPR in cancer treatment. In 2019, scientists in the US and China used CRISPR to edit the immune cells of cancer patients, helping their bodies better recognize and fight off tumors. These early clinical trials focused on modifying T cells to target specific types of cancers, such as lung cancer and multiple myeloma. Another study used CRISPR to disrupt a gene called PD-1 in T cells. PD-1 is a checkpoint that normally prevents T cells from attacking the body's own cells, including cancer cells. By editing out PD-1, the modified T cells became more aggressive in attacking the tumor. Challenges and ethical concerns. Off-target effects. One of the main challenges of using CRISPR in cancer treatment is the possibility of off-target effects, where the CRISPR cuts DNA in the wrong place, potentially causing unintended damage to healthy cells. Ethical considerations. Editing genes, especially in ways that affect the entire immune system, raises ethical questions about long-term safety and unintended consequences. Researchers are working hard to ensure that CRISPR treatments are both safe and effective before they become widely available. In summary, CRISPR is offering new hope in the fight against cancer by allowing doctors and scientists to directly edit the genes of cancer cells or enhance the body's immune response to target tumors more effectively. From enhancing immune cells in CAR-T therapy to disabling genes that drive cancer growth. CRISPR's potential in treating cancer is vast. While there are still challenges to overcome, the progress made so far suggests that CRISPR-based cancer therapies could become a key weapon in the battle against this disease in the future.